Enterobacter, and others. However, the bacteria that were retained within the Erwinia genus were delineated into 44 distinct species, among which there are only a few plant pathogens. And if we zoom in a little bit, these plant pathogens, most of the majority of these plant pathogens cause necrotic disease. And Erwinia tracheiphila is the only species within the Erwinia genus that causes that causes a vascular disease. Erwinia tracheiphila is obligately transmitted by cucumber beetles, which could either be striped or spotted, and it's a xylem restricted bacterium. It is the causal agent of bacterial wilt of cucurbits, and the diagnostic sign of this infection are these slimy bacterial strains, which you see when an infected plant is cut at the crown, and then you just kind of gently, pull it apart. This disease is endemic in the eastern half of the United States, where losses can exceed 80% on cucumbers and, and melons, with a potential annual loss of more than $20 million. The beetles survive the winter as adults, and then they become active in the spring and feed on young susceptible cucurbites, during which bacteria infested pulp, contact wounds, and flowers, leading to the movement of the bacteria into the host xylem, and then the bacterial cells multiply. They block xylem water flow, which ultimately results in the characteristic wilt symptom, and then eventually the plant collapses and dies. Because of a lack of genetic resistance to bacterial wilt, management of this disease has largely depends on frequent insecticide applications to kill the cucumber beetles. Strains of Erwinia tracheiphila exhibit differences in host-specific virulence. Here, we are looking at two different hosts, squash and muskmelon. What we have seen so far is that some strains rapidly with squash and they cause slow wilt on muskmelon, in which case the squash is a primary host and muskmelon is a secondary host. So based on the ability to cause rapid wilt on squash versus muskmelon, we could say these strains are squash strains. Whereas some strains rapidly wilt muskmelon but are unable to wilt squash. And also some strains, they're unable to wilt squash. And also we could say the strains are, based on ability to rapidly wilt muskmelon, they are melon strains. We also do know that the Erwinia tracheiphila strains clustered into three phylogenetic clades with distinct geographic distributions based on similarity across the genomes. And the Et-melo clade that we can see here, they're exclusively the melon strains. Remember the melon strains, they cause rapid wilt on melon, and then the squash strains cause rapid wilt on squash. Whereas the Et-C1 and the Et-C2 clades contain the squash strains. They contain the squash strains. And interestingly, surprisingly, all the strains in this study were actually located only to one region of the world, and that's the United States and the southern Canada. And among these, the Et-melo strains are the most broadly distributed, as they do cover the entire range. But the Et-C1 strains only occupy the eastern part of this range. These genetically distinct clades vary in their Type III Secretion System effector repertoire. Effectors encode genes that enhance the virulence and fitness of many gram-negative phytopathogenic bacteria, and they are secreted to their plant targets through a complex system that is known as the Type III Secretion System. Despite the pathogenicity and the genetic dichotomy among Erwinia tracheiphila strains, we still do have some knowledge gaps that have limited our understanding about the pathogen system. And this led us to the goal of this project, understanding the genetic basis of Erwinia tracheiphila speciation and its association with host-specific virulence. And just as this, as the biblical scripture that says that why do the nations rage and the people imagine the vain thing? We're just like, why are the melon strains so, so angry that they just want to get rid of all the muskmelon, in the United States? And also, why are the squash strains? Why are they just so angry against the squash, against the American squash? So that's the goal of this project. So we're going to be addressing these questions using molecular tools. So to address this goal, we came up with certain questions. The first question is, are the Erwinia tracheiphila effectors expressed in a pattern similar to the observed or specific virulence? The next one is, do these effectors even have a role in or specific virulence and pathogenicity? Are these effectors, are there effectors that primarily contribute to the virulence of Erwinia tracheiphila clades on their respective cucurbit hosts? And is there an association between the Erwinia tracheiphila type III secretion structural genes and clade differences in host specificity? And lastly, are the clades different enough to warrant a subspecies delineation among Erwinia tracheiphila strains? Now let's begin to address these questions one after the other. Based on the reported clade designation, we base our studies on the Et-melo and the Et-C1 clades. So those are the melon strains and the squash strains. We did not include the strains of the third clade, which is the Et-C2, because we do not have access to those strains from the clade, from this strains from this clade. So for each clade, what we did was to assemble the genome sequences that included the PacBio generated long read sequences, which were used as reference for 11 Illumina generated short read sequences. And this makes a total of 12 strains. It makes a total of 12 strains per clade. Excuse me, I'm trying to do something on this. Just trying to get the, okay, so let's just move on. Okay, so using a local database, a local blast database, we mine the effectors from the representative strain of each clade, and now we identify 17 effectors. A majority of these effectors are shared by both clades, and 3 of them are actually specific to each clade. So we wanted to know what effector genes are expressed by the melon strains on its host. And also the melon strain also does, although does not induce wheat on squash, but we found out that at least 2 strains replicate pretty well within squash. So we were curious to see if there could be something interesting about the expression profile of those effectors in squash. And of course, which effectors are expressed by the squash strain on its primary host squash and secondary host muskmelon. So how did we design this experiment? We inoculated squash and melon seedlings, each with melon strain, and then did the same thing with squash. We went out to collect xylem sap, and then from the xylem sap, we extracted RNA. From the RNA, we evaluated transcript abundance using QRT-PCR. Using QRT-PCR. So among the effector genes we examined in the melon strain, what we actually found out was that Eopt1 was the most highly expressed. And this Eopt1 was actually most highly expressed in melon, and interestingly also on its non-host squash. Although we had additional genes that were being expressed, I'm just gonna leave this here for now, because those names could be very, very confusing. Just trust me. I've fought with this for a while, but I'm just gonna make a reference to this as we move on in this presentation. Interestingly, Eopt1 was also the second most highly expressed effector gene in squash strain on both its primary and its secondary hosts. But in addition to the Eopt1 gene, we also realized that there was this particular effector gene that's called HopL1. This gene is actually present in squash alone. It's present only among the squash strains. What we actually found out was that this gene was expressed at an exceptionally high level. Again, I'm just gonna stop right there for now. So let's just kind of really keep it simple. So let's move on. So remember, EOP1 was expressed was expressed in both strains on both O's on their respective O's, and also O1 was expressed on only in the squash strains in both O's. So at least that kind of gave us an idea of the expression profile of tracheiphila strains and their respective O's. So it guides us to know which effectors are actually worth exploring in terms of their functions. Based on expression profile and reported roles in host specificity among some phytopathogenic bacteria, we selected EOP1 as a putative candidate for host specific virulence in Erwinia tracheiphila. And if you look at this publication, it's actually showed that EOP1 limited the host range of Erwinia amylovera strains. And so they had these berry strains that exclusively infected berries, but just won't infect apple and pear. So when they knocked out or deleted this particular EOP1 gene from the berry strain, it gained the ability to cause disease on apple. So which means that the presence of EOP1 in the berry strain actually limited its ability to cause disease on apple. So when they also moved, which was a kind of overexpression, when they overexpressed the EOP1 from the berry strain and the apple strain, it also reduces virulence. So they concluded that EOP1 from the berry strain actually functioned as a host limiting factor. So therefore, we hypothesized that EOP1 perhaps has a role in the virulence of Erwinia tracheiphila. We further found that EOP genes are highly conserved within the Erwinia genus. Like we can actually see there, and that EOP1 sequence is identical within each clade, but differ by 12 nucleotides between the two clades. And that these are the no, those are the two, those are the two, those are just two out of the 12, those are two out of the 12 SNPs. And a single nucleotide polymorphism have actually been shown to drive host specificity among plant pathogenic bacteria strains. We further found that EOP1 genes are highly, okay, so let's just move on. Okay, so despite this high expression level, what we actually found out was that deleting EOP1 did not alter its virulence at all on both clades, on both O's. So what you're actually seeing on the left panel is the wilt. That's the width progression, and on the right panel is the area under the cis progress curve for the same data. So this is the result from two melon strains. The loss of EOP1 did not cause these melon strains to width squash as well. Similarly, within the squash strains, loss of EOP1 had no phenotype. So these findings indicate that in contrast to its role in Erwinia amylovera, EOP1 is not a host limiting factor in Erwinia tracheiphila. And because its loss did not alter virulence on respective hosts. However, this is not uncommon with effectors, as the loss of one among several rarely confers any phenotype. So this is often due to functional redundancy, in which case effectors have overlapping functions, meaning that one is compensating for the loss of the other. Sometimes overexpressing a gene either in its native bacterial host, which is homologous expression, or in a different bacterial host, which is heterologous expression, confers some virulence or avirulence traits. But in the case of this study, neither of these expressions alter the virulence of the overexpressed constructs on their respective primary hosts. So this is a melon strain of melon, and then the squash strain on the squash strain of squash. We did not see any phenotype. However, when we heterologously express Eopt1 from a melon strain in a squash strain, it also did not alter the virulence on primary host, but it increased the virulence on the secondary host muskmelon. Remember that the squash strain induces lower weight on muskmelon, but overexpressing the Eopt1 from a melon strain, which is this one, the one that's number 4 here, actually increase the virulence compared to the white type. So we confirmed our findings with a QRT-PCR expression assay using xylem sap extracted from infected plants. And it actually showed that we had implanter expression in our construct. So these findings actually demonstrate that Eopt1 functions it functions in host-specific virulence. That is virulence that is specific on a muskmelon host. Although DSP was barely expressed in both melon and squash strains in both respective hosts, it is an important pathogenicity determinant among many plant pathogenic bacteria, including Erwinia millavora, Pantois stewarti, Rastonia solanisterium, and Pseudomonas syringae. It's a big chain, which is about 5 kb, and similar to Eopt1, it is highly conserved within the Erwinia genus. So we explored this gene as a pathogenicity candidate. To fish for genetic basis for pathogenicity, we deleted DSP and its chaperone in selected melon and squash strains. A DSPE deletion method reduced the virulence of 2 different melon strains on muskmelon. The remaining, so we can actually see here that this is a melon strain. When we deleted the SP, you can see the rates of of reduced virulence in both melon strains, in these 2 melon strains. So this is actually telling us that this particular we still have some virulence, some level of virulence left in this particular strain. So the remaining virulence actually demonstrate that DSP is not absolutely required for the virulence of the strains. Actually, this rule was predicted because of that's because of the contribution of DSP family members to the pathogenicity of many plant pathogenic bacteria. Surprisingly, this rule was different among the squash strains, as the loss of DSP completely eliminated wilt induction on both primary host squash and secondary host muskmelon. And this is actually the first squash strain, and also we found the same thing in the second squash strain. So as a summary, Erwinia tracheiphila is the only plant pathogenic species that includes strains that have different roles for an AVRE member. The other strain, so we can actually see that that's the those are the melon strains, and then the ET and then the squash strains. The others occurrence that we've seen so far was actually within the Pectobacterium genus, and this was actually at the subspecies level, and not at the species level like what we actually see with Erwinia tracheiphila. So the take-home message for all I've said so far is that Eopt1 functions as a host-specific virulence, and also that the DSPE is diverse in its function between the two groups of Erwinia tracheiphila strains. So we went ahead to look at it that to look at the next question, that are there effectors that primarily contribute to the virulence of Erwinia tracheiphila clades on their respective cucurbit host? Based on expression profile and lack of phenotype in Eopt1 deletion mutants, we began to explore the function of other effectors. And for simplicity's sake, this figure is just going to serve as a roadmap for my presentation in this section, so that we don't have to begin to memorize, the names of these effectors. So we use a combinatorial mutant analysis to characterize the clade-specific roles of these effectors. So the very first one we found was that a deletion mutant of AVRB4 in a melon strain reduced its virulence on its muskmelon host. So this melon strain, when we deleted AVR this AVR was one of the effectors that was expressed here um, in the melon strain that we used in this study. So this actually demonstrates that AVR contributes to the virulence of this particular strain. The loss of OspG alone did not affect virulence, but the loss of this effector in combination with Eopt1 resulted in reduced virulence. So we suggest that OspG is one of the effectors that's probably compensating for the loss of Eopt1. We evaluated the quantitative contribution of the 4 melon strain effectors to virulence using pyramided mutants. And what we actually found was that the major effectors that had measurable individual contributions to fitness were only AVRB4 and DSP. And the loss of the SPE together with the chaperone actually had the largest impact on virulence among these strains. So if we back up a little bit to our Venn diagram, we can see a bigger picture of the effector repertoires and how their relative contributions was kind of seen to the virulence of each of this clade. So the major effectors that had measurable effect, measurable contributions to fitness of the melon strains were AVRB4 and DSPE. With the loss of the SPE, the effects of Eopt1 and OspG were only detectable in the absence of other effectors. So from here, we just kind of moved on to the squash strains. So this is a story about the melon strains, the effectors that are critical or that contribute, to their virulence. So now we move to the squash strains. Interestingly, despite its exceptionally high expression level, the loss of HopL1 did not alter virulence of the squash strain on its primary and secondary hosts. Remember, this is HopL1. It was expressed at an exceptionally high level. But if you look at it, when we deleted this particular gene from HopL1, it did not influence the virulence on both melon and squash. And we found the same thing with HopL1, which was also highly expressed in squash strain on its secondary host, and did not alter virulence at all as well. We also found the same thing with OspG. So again, this is not uncommon. When you delete a particular effector gene, you don't get a phenotype. This is just what we're seeing here. So nothing strange for now. Let's move on. So we began to identify some effectors that have overlapping function with Eopt1. And among them are HopL1 and HopL1. So although their individual losses did not alter virulence, but their losses in addition with Eopt1 reduced virulence on muskmelon. So this is HopL1. If you can see this green bar, so these two green bars for HopL1 and for HopL1 was when we deleted both Eopt1 and HopL1, and Eopt1 and HopL1 in each case. We notice a similar Eopt1 functional overlap with HopL1 and HopL1, 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 and also OspG in squash strain, but this time on its primary host squash. Among them, HopL1 and HopL1, that's what we kind of found. Although their individual losses did not alter virulence, but their losses in addition with Eopt1 reduced virulence, and that's what we also found with HopL1, HopL1, and also OspG. Similarly, we evaluated the quantitative contributions of these four effectors, Eopt1 AVRB4, OspG, and DSP, that's the squash effectors, on the virulence using pyramidal mutants. And what we actually found was that the combinatorial loss of all these four effectors had a great impact on virulence among these effector strains. So as a summary, looking at the big picture, the effectors that had the most critical impact on virulence of the squash strains, remember, is DSP. That was one that, when we knocked it out, the strain became completely non-pathogenic. And the effector that adds, whereas effectors like HopL1, Eopt1 and OspG and HopL1 appear to have some overlapping functions. So the take-home message for this part of my presentation is that the two Erwinia tracheiphila clade strains rely on overlapping but distinct set of effectors for virulence. And these are the list of effectors that are critical to the virulence of the melon strain and also of the squash strains. And an addictive effect of the loss of the selected effectors were also detected in the strains of both clades. So let's move on to the next question. Is there an association between the Erwinia tracheiphila type III secretion structural genes and clade differences in O specificity? Erp-A is the major component of the type III secretion system pillars. And this gene is highly conserved among plant pathogenic bacteria. And usually the loss of this gene usually makes a phytopathogenic bacteria lose the ability to cause disease. So if you look at it, this is like a pipe, it's like a straw that is required by the type III secretion system to secrete effectors into the O cells. So if this particular pipe is missing in plant pathogenic bacteria, it prevents the ability of those effectors to be secreted into the O cells. So for the expression studies, what we did was to use Erp-A as a control gene for expression of the type III secretion apparatus itself. And the expression of Erp-A in a melon strain in muskmelon was more highly expressed in muskmelon than in squash. So this is a melon strain expressing Erp-A more on its host melon, and it was barely expressed on squash, which was a non-host. And also, if you look at the squash strains, the squash strain actually expressed Erp-A on both squash and muskmelon. So, as expected, the loss of Erp-A in a melon strain, made it lose pathogenicity on muskmelon. Again, I'm just going to use the expression data as a roadmap for ease of comparison in this presentation. Similarly, as expected, the loss of Erp-A in a squash strain made it lose pathogenicity on muskmelon. So these findings tell us that not only are the type III structural genes expressed in plantar, but they are also active among the Erwinia tracheiphila strains. There's a positive correlation between Erp-A expression levels and those of the cognate type III secretion effector genes. And this was very similar to actually this has been reported among the Pseudomonas syringae pathovars. So based on this, we hypothesize that a quantitative overexpression of Erp-A in the melon strains will probably cause it to wheat squash. Remember, the melon strain does not wheat squash at all. And again, if you look at the expression, its expression here, if you look at this expression in the melon strain on squash, it was barely expressed. So we hypothesize that if we try to overexpress this gene here in the melon strain, it's probably going to gain the ability to wield squash. Interestingly, consistent with our hypothesis, all the melon strains that overexpress Erp-A wilted squash, albeit at significantly lower rate than BHKY. So if you look at, let me just kind of walk you through this really quick. So this is BHKY. This is a squash strain that's that with squash. But if you look at this, the buffer and also the SL3 is just as there, so it doesn't cause any weight on squash. But if you look at from number 1 to 5, these are the constructs of SL3 that are overexpressing Erp-A using Erp-A that was fused to different promoters, and all of them actually showed some significant levels of wilt on squash. So this is actually the very first time we're going to be finding melon strains getting the ability to watch squash. So we did an implanted expression profile of our constructs and found that an overexpression of A variant in the melon strain is associated with observed ability to weight squash strains. So if you look at it, so we did both in vitro. If you look at the red bars, those are the in vitro expression assay, and the orange bars are the implanter. So what we actually found out was that not only were these constructs able to weight squash, but they were also the A level of expression within them were higher when we looked at them implanter. So the take-home message is that the tracheiphila Type III Secretion System is expressed in squash and also in muskmelon. And this Type III Secretion System is active in the translocation of the cognitive effectors. And also that tracheiphila clade differences in host specificity are associated with host specific virulence differences in A expression. So finally, we addressed the question of, are these clades different enough to warrant a subspecies delineation among the older tracheiphila strains? To address this goal, so what we actually did was to look at the differences that were enough to probably say, okay, these strains are different enough. In the process, we can probably try to put them, delineate them into distinct subspecies. So based on that, we looked at predictive functions. So what we actually did was to conduct a kind of pangenome analysis on all the 24 strains, and we looked at predictive functions, including toxin antitoxin modules, prophages, and then the carbohydrate active enzymes. We also looked at differences in plasmic content, differences in physiological traits like carbon substrate utilization profiles, and also in vitro growth rates. And also we looked at differences in virulence traits. And that was actually virulence on muskmelon because both strains cause disease on muskmelon. So let's look at what we found. Sorry, my computer is trying to hang. Okay, there you go. So we observed that division of all 24 strains were actually, we observed the division of all 24 strains into two separate clades, which was very similar, consistent with what we've seen before. But we actually found there was that we found a kind of low genetic diversity within the strains. So what we're trying to see here is the red rings are the squash strains. The red rings, they are the squash strains, and the blue rings are the melon strains. So on the left panel, all the strains were aligned against a squash strain, and this showed the degree of differences within the melon strains. And this is actually, denoted. This is denoted by the variable region. So we have about 3 variable regions among the melon strains, and when we did the same thing for the squash strains, we found just one variable region within the squash strains. And interestingly, if you look at this particular finding, this was actually despite the fact that all these strains came from throughout a large region. For can see, these are the Et-melo, these are the melon strains, these are the squash strains. So despite the fact that they came from, throughout a large region, we were able to find this level of diversity, genetic diversity within the strains, within each clade. What we also found was that the melon strains have twice as much as the TA modules. So the TA modules are the toxin antitoxin modules. And what we actually know about that is that this particular loci, when they are present in plant pathogenic bacteria, they function as a feedback system in response to environmental and nutritional stress. So we do not know the benefits that these, having as twice as much as the toxin antitoxin modules within, in the among the melon strains, we do not know the kind of benefit it probably confers on the strains. One of the striking findings we actually found from the comparative phage profiling is that Among the 83 total prophages that were identified in this study, only two only two of those strains are shared among these 24 strains. So each clade has its own distinct set of prophages. So a majority of the identified prophages we found were MU-like in the families of Podoviridae, Cyphoviridae, Myoviridae, and Inoviridae, which are the filamentous prophage. So the MU-like phage, they are known to encode virulence genes or induce host expression of virulence genes. So whether these identified prophages also contribute to the virulence of tracheiphila strains remains unknown, and this will actually be worthy of investigation. CAZymes those are the carbohydrate active enzymes. They are responsible for the synthesis and breakdown of polysaccharides, especially from bacteria. These bacterial CAZymes, they are present and they can actually break down all these all these polysaccharides. So as a xylem inhabiting bacterium, the intervessel movement of Erwinia tracheiphila requires passage through the pit membrane, and polysaccharides present in the pit membranes are therefore potential substrate for carbohydrate active enzymes. So if you look at this, these are xylem vessels. So when a tracheiphila gets in air, and the xylem being very poor in terms of nutrient, um, it's going to require this tracheiphila strains to move from one xylem vessel to another in order to maximize their access to nutrients. So for them to be able to do that, these pit membranes are going to be they're going to be barriers that they're going to have to go through, to overcome. So these barriers have polysaccharides, and we're it's possible that these polysaccharides um, they are often the targets of these carbohydrate active enzymes, like we found in some xylem inhabiting bacterium. So let's see, what do we do? What do we do with this? Most of the differences we actually found. Okay, my computer is silent. So most of the differences we found between these two clades were actually within the glycoside hydrolase family of enzymes. So the glycoside family of enzymes, and the most important of this glycoside hydrolase is the GH6. This has actually been well studied. And we actually found was that there was an overrepresentation of the GH6 subfamily of the glycoside hydrolase among the melon strains. So what we had to do was to zoom in a little bit to the GH6 to see if we could find something interesting that shows differences between the two clades. So interestingly, what we actually found was that we found a GH6 member, which is a cellulose gene. And this gene was only present among the melon strains, but absent of the squash strains. So interestingly, this gene has a critical role in differentiating vascular from the mesophyll infections on bacterial plant pathogens. In other words, when they move this particular gene from a non-vascular bacterium into when they moved into a non-vascular bacterium, it became a vascular pathogen. So what we did was to delete this particular cellulose gene, and interestingly, when we deleted this particular gene, this cellulose gene, it reduced the virulence of the melon strain on its host, muskmelon. So which kind of tells us that this particular gene contributes to the virulence of the melon strain. Remember, this gene is absent in the squash strain. So we also move this gene into the squash strain to see what's going to happen, if it's going to gain the ability to probably wield muskmelon or squash faster. But surprisingly, what we found was that when we moved this gene into the squash strains, it reduced the virulence of these strains, of the squash strains, both on its primary and on its secondary host. So this actually kind of tells us that each clade is very specific in a selection of subsets of carbohydrate active enzymes, and that the enrichment of the GH6 CAZyme family appears to play a role in the virulence of the melon strains. When we looked at the differences in when we looked at the differences between the plasmid content of these strains, what we actually found was that most of the melon strains had multiple light plasmids. They had this multiple light plasmids, which are about 40 kb, and also medium between 10 to 40 kb, and also some smaller plasmids. In contrast, the squash strains generally contained just between 2 to 3 plasmids. So again, this kind of tells us, was a major difference between these two strains. And consistent with the differences in the carbohydrate active enzymes accumulation between both strains, what we actually found was that each clade actually differs in its carbon utilization profile. Although they are able to utilize about 10 identical substrates, they have preference for distinct clade-specific substrates that we can actually see here. And what we also found was that the squash strains grew slower on average than the melon strains. And also that, this is consistent with what has actually not been found, that the squash strains actually induce wilt slower than the melon strains on melon. So these were all the differences we actually found between the strains, in these two clades. So on the basis of the data from growth rates, carbohydrate utilization, genomic architecture, and pangenomic features, we propose that the two clades of Erwinia tracheiphila previously designated as Et-melo and Et-C1 clades be designated as two subspecies. And what are these two subspecies? The Et-melo strains be designated as Erwinia tracheiphila subspecies tracheiphila. And this is based on convention in which the first subspecies is given the species name. And the Et-C1 strain, which is Erwinia, renamed as Erwinia tracheiphila subspecies lentillata. And the lenti is actually from lenti and lata. They are from Latin words which means slow and broad, respectively. With that, I come to the end of this presentation. I just want to say thank you to my committee members, especially going for our mentorship role in getting this project done. And I just really want to say thank you very much. And also to the BT and Harvesting Lab. It's been great getting to know, and interact with you all. I really love you and appreciate you all. And Benzong was really helpful in all the gazillion plant studies that I did, and Chen too. So constructing, trying to genetically manipulate Erwinia tracheiphila was very, very challenging when I got to Gwen's lab, and Chen was really helpful at those early stages, and I really appreciate that. And also thank you to my funding sources, and also I won't go without saying thank you to my family, my wife especially, for her support and sacrifice since this past 6 years, and thank you very much. And my lovely kids, Michelle and Joshua, they are very good at keeping that complaints. So thank you very much. And with that, I would like to take some questions.
Thank you very much. In a normal situation, we have a round of applause. So applause. We'd like to be able to open it up for any questions that anybody has for Kunle. I'm going to start by asking you to explain why lentillata. Slow is growth rate, but what do you mean by broad?
Okay, thank you. So lentillata is, if you remember the geographical the map that showed the geographical distribution of the strains, there was ETC1, there was ETC2. So the ETC1 was more distributed, was more broadly distributed than the ETC2 strains. So because of that, that's where the word broad actually came from. Then lenti is actually from the fact that these strains, they are slower growing than the melon strains. So that's than the Et-melo strains. So that's where the slow and the broad actually came from.
Thank you. Do we have other questions for Kunle? Quiet group today.
I have a question. So Kunle, can you talk at all about the importance of EPS production for the pathogenicity of these strains? And do you have any indication that the EPS differs between them?
That's a very interesting question. So about the EPS, we do not have any information about the EPS, about the role of the EPS in the pathogenicity or virulence or relative virulence of these strains. But what we do know is actually that, except that when they get into the xylem, they kind of, they multiply, they produce a lot of, extracellular polysaccharide, and in the process, all the APS that are able to, they kind of plug, the xylem flow of water and nutrients, and which eventually results into to the wilt symptom. So we do not have the EPS of tracheiphila. We do have evidence though that the genome actually has some extracellular polysaccharides, and but it's not been characterized. So I think that will be something that will be probably worth looking into in the future.
Okay, so just to make sure I understand, you have those few, the few melon strains that can actually infect squash, but they don't cause wilt, right?
Yes.
So they're able to reach the xylem and they just never produce the EPS to basically block that flow, or are they replicating somewhere else? They're replicating in the xylem, right?
Yes, they replicate within the xylem, but yeah, I think that's a very interesting question. So they replicate in the xylem, but we do not have evidence yet to support their relative production of EPS within the xylem. So the information we have so far is that when we inoculate the squash, when we challenge the squash with the melon strains, they are able to replicate within the host. So by just kind of collecting xylem sap, and we see a lot of the melon strains within the squash. But why they do not collect, why they don't cause infection, probably relative to whether they produce EPS or they do not produce EPS, it's not something we know at the moment.
Okay, thanks.
We know that they're not getting the effectors over because the HARP-A gene isn't expressed. They're not getting their Type III secretion system. But when you get that expressed, if the polysaccharides are kicking in, they're not taking it all the way there. Any other questions for Kunle?
I have a question, Kunle. And years ago, they used to use pathovar designation for a species that had different pathogenicity on different hosts or some other pathogenicity factor. Is that still used in bacteriology today? And how many pathovars are there of tracheiphila?
Thank you very much for that question. Yes, absolutely. They still use pathovars, and that's very common within among the Xanthomonas, species, and Xanthomonas and also and Pseudomonas. But with Erwinia tracheiphila, we do not have any pathovars for now. And that's one of the things we're kind of really trying to see, that do we kind of really delineate them as subspecies of pathovar. But with pathovar is probably not as relevant to these strains, because pathovar is actually more of it's kind of more genetic. So we probably don't have enough information to kind of really look at enough information about the host pathogen interaction at the molecular level to help us warrant the ability to go ahead and say, okay, these strains are probably going to be delineated as pathovars. So There are no pathovars for Erwinia tracheiphila, but we are proposing a subspecies delineation based on the differences we currently have.
Okay, thanks.
Generally, like with Pseudomonas and Xanthomonas, when you have a pathovar, the main difference you know about is the host pathogenicity. And that often is the only difference and is probably tied to effectors. Whereas here, it seems like there's so many other things that are also different between the subspecies, and the pathogenicity is overlapping on hosts. So as not the primary determinant, it seemed like a poor name. I would add that. Any other questions? Well, if not, then I hope you can join me in giving a final round of thanks to Kunle for a nice presentation. And I'll let Leonor do the closing here.
Thanks y'all for coming, and congratulations Kunle for your seminar, and
Yes.
we'll
Thank you.
see you all next week.
Thank you.
Thank you.
Bye.